Today we will be discussing about mode of action of various antiviral drugs, various antifungal drugs, various antiprotozoal drugs and anti-Helminthic drugs. So let's start with antiviral drugs. If we look into the mode of action of various antiviral drugs, they can be broadly divided into 4 different categories. Number 1 category is those drugs that prevent the uncoating of the virus, or they prevent the entry of the virus inside the host cells. Second category of antiviral drugs are those that prevent the nucleic acid biosynthesis pathway of the viruses. Third class of antiviral drugs will be those that prevent the integration of the viral genome into the host genome. This will be specifically important in case of viruses like HIV or retroviruses, wherein we can see the integration of the viral genome inside the host genome. And fourth class will be those antiviral drugs that prevent the exit of the viruses from the host cells and prevent their reinfection pathway. So let's start with the antiviral drugs that prevent the entry of the viruses inside the host cells. The first member of this group are called infuverdides. They are the entry inhibitors. What they do? They bind to those proteins, specifically the HIV virus proteins are being blocked that are important for the fusion of the virus with the host cells. So one of the important such drug is called Maraviroc. It binds to the co-receptor called CCR5 and prevents the fusion of the virus to the host cells. Adamantins comprises of amantadine and rimantadine. These two drugs are specifically used to combat influenza virus infection. What they do? They interfere with influenza virus ion transport mechanism, and they are inhibitors of hemagglutinin receptors. Thereby, they inhibit the fusion of the influenza viruses into the host cells. So these drugs are only active until active immunization can be achieved. Unfortunately, resistance develops frequently and limits the usefulness of these drugs. Next we come to the nucleic acid biosynthesis inhibitors. In this class we can start up with purine pyrimidine inhibitors or analogues. What we do, several purine and pyrimidine analogues are used as successful antiviral agents. Here what is done, that these analogues are getting phosphorylated by viral enzymes or cellular enzymes, and they prevent the incorporation of normal nucleotides. Because of the incorporation of these analogues, what happens that there is premature termination of DNA synthesis, or in some cases we may see that this incorporation lead to a mismatch in the DNA, which leads to finally destruction of the DNA.
Ma'am, how selective toxicity is achieved in case of purine and pyrimidine analogues?
If we are using this growth factor analogues or this purine pyrimidine analogues, then it might affect our DNA synthesis mechanism. But the answer is that the viral coded enzymes which are responsible for the synthesis of viral DNA synthesis, they have much more affinity towards these analogues rather than our own DNA polymerase. Means our own DNA polymerase has got lesser affinity towards these analogues. And second reason is that many a times the drugs that we are administering is administered as a prodrug. Once it gets inside the host cell which is infected with the virus, then the virus will start producing certain enzymes which will modify the drug into a real analog, growth factor analog or purine pyrimidine analog. So the drugs that we are administering are not active until and unless they are getting modified by viral coded enzymes. So let's start with purine pyrimidine analogues that are used as antiviral agents. We start with idoxuridine, which is an analog of thymine. It is used for the treatment of eye infections caused by herpes viruses. Next we have got vidarabine, which is an analog of adenine. It is used for the treatment of encephalitis caused by viral herpes. It causes damages in the brain and it is effective to treat the herpes virus infection. Acyclovir is a growth factor analog, or it mimics guanine residues. It is used for the treatment of various varieties of herpes infections as well as cytomegalovirus infections can be cured taking help of acyclovir. Ganciclovir is also an analog of guanine, but it is better than other antiviral agents. Next, we have got zidovudine, which is reverse transcriptase inhibitor, and it is used for the restriction of growth of retroviruses.
Ma'am, which one among all these nucleic acid biosynthesis inhibitors, which antiviral drug is possessing least toxicity?
If we consider about the toxicity criteria, then acyclovir is supposed to be the best drug. Reason behind is it is administered as a prodrug, and the conversion of the prodrug into drug is being done by a viral coded enzyme and not by host coded enzyme, as a result of which only those cells which are being infected by the virus, and as soon as the drug gets inside those cells, then only it is becoming active or effective. Those cells which are not infected with viruses, therein even if the drug gets access, it is not acting as an analog because it remains as a prodrug. That's why we consider acyclovir is supposed to be the best drug among the purine pyrimidine analogs. So in this category of nucleic acid biosynthesis inhibitors, we have got another class of antiviral agents that are called non-nucleoside polymerase inhibitors. These enzymes actually inhibit the viral polymerases by binding to the sites which are different from the nucleotide binding site. Phoscarnate is one such drug which is used under those cases wherein herpes simplex virus is showing resistance against acyclovir, or cytomegalovirus is showing resistance against some of the drugs. Then we use phoscarnate. Next category of drugs within this nucleic acid biosynthesis inhibitors are reverse transcriptase inhibitors. In this case, we will see that the drugs are inhibiting the reverse transcriptase activity of AIDS virus or HIV virus. Nevirapine is example of this category. The third category of antiviral drugs that we are about to discuss are inhibiting the integration of the viral genome into the host genome. In this category of drugs, we will be specifically focusing on the drugs that target the HIV virus. They prevent the integration of the viral genome into the host genome. Raltegravir is an example of this category. Finally, we are coming to the category of antiviral agents that prevent the coming out of the virus from the host cells, or the exit of the viruses from the host cells. In this category, we will be discussing about the mechanism of assembly of the viruses before their exit. So a virus, if it wants to come out of the host cell, before that it has to synthesize a large number of proteins, and many a times it synthesizes the proteins as polyproteins. So these polyproteins are to be cleaved by certain proteases. These proteases are also viral coded proteases. Many drugs are there that inhibit these proteases and prevent the cleavage of the polyprotein into functional protein molecules. This type of drugs are specifically used in case of HIV viruses, wherein what they do, that they synthesize proteins as polyproteins. Neuraminidase is an enzyme which is needed for the exit of influenza virus from the host cells. So, neuraminidase inhibitors will inhibit the action of neuraminidase, thereby they can prevent influenza A and influenza V. The drugs in this category are oseltamivir and zanamivir. Oseltamivir is popularly known as Tamiflu. It is used for the treatment of influenza A and B. And oseltamivir is also used prophylactically, as well as it can be used as a drug, whereas the other drug which is used only as a drug but it cannot be used as prophylactic action. Now we will be discussing about the drugs that induce immune responses. So, levamisole is a drug which is administered to induce the T leukocytes. Thereby, we can have our own immune response active to take care or combat the viral infections. Induction of interferons or production of interferon is a type of defense mechanism that is operative inside our system to combat any viral infection. So, the cells which are infected with viruses start producing interferons, and these interferons will then infect the other cells, and they will make the cells resistant to infection towards viruses. Now what we have done, that we have started generating some genetically engineered compounds which can enhance this release of interferons, or which can act as inducers of interferons, and thereby we can prevent the action of the viruses. Development of chemotherapeutic drugs against funguses poses similar problems faced while developing drugs against viruses. Reason behind that funguses are belonging to the same group Eukarya, as a result of which whatever pathways that we are to choose are also present within the host cells. That's why antifungal drug designing is becoming a difficult thing. What we have seen here, that we have chosen those target sites within the fungus structure which are absent in the host cells. And secondly, the development of antifungals are becoming important nowadays. Reason behind that immunocompromised patients or patients who are suffering from AIDS are very much susceptible to infection with the funguses. Ergosterol is the replacement of cholesterol which is present in our system. So, ergosterol is an important sterol present in the fungal membrane. It can be disrupted by a class of antibiotics and antifungals which are called as paulines or allylamines, and we start with paulines. Paulines comprises of ability to bind to the ergosterols, and binding to the ergosterols, they generate transmembrane channels. Through these channels, monovalent ions like potassium, sodium, H+ ions come out of the funguses, and this leads to the disruption of the fungus and finally leading to death of the funguses. And category of drugs within this pauline are nystatin and amphotericin. Amphotericin B and nystatin, both of them are useful in treating infections caused by Candida albicans, or they can be used to treat infections like aspergillosis. Azoles and allylamines. Azoles comprises of those drugs like ketoconazole which are used for superficial applications like skin eruptions in those places or some infection in the eyes, wherein we can use ketoconazole, clotrimazole. but they cannot be used for internal uses. The reason behind is they cannot enter inside the animal cells. The next important target site for antifungal drug is echinocandins, which is the fungal cell wall unique component. It comprises of polymers of glucan, which is synthesized by an enzyme 1,3-beta-D glucan synthase. So echinocandins are targeting this as their drug target sites. They bind there and they prevent the synthesis of fungal cell wall. The only echinocandin that is approved by FDA is caspofungin. It is used for the treatment of aspergillosis and candidiasis. In the next category, we have got flucytosine, which is a very good antifungal drug. It is an antimetabolite. What it does, it actually interrupts the nucleic acid biosynthesis in funguses. Finally, we do have Griseofulvin. It is also an antifungal drug which actually prevents the mitosis events by binding to the microtubules. It binds there and prevents the mitosis event which is going on within the funguses. As compared to antifungal, antiviral and antibacterial agents, very little is known about anti-parasitic agents. The reason behind is these type of infections are mostly caused in those countries where people have got little resources or little money to do research on this area. And parasites being again eukaryotes, it is a bit challenging to design anti-parasitic drugs. So specifically, we will be choosing only those unique metabolic pathways which are undergoing within the parasites which are not present in the hosts, and we are targeting the neuromuscular functions of the parasites. It is supposed to be the first drug that has been isolated from the barks of cinchona tree. Now that these drugs are very often grown in Indonesia. Chloroquine prevents the action of Plasmodium falciparum or Plasmodium vivax. How it executes its action? That it executes its action by preventing the breakdown of hemoglobin. Primaquine is used prophylactically to treat those persons who are susceptible to infection by this Plasmodium vivax or Plasmodium falciparum. So it is used in combination with chloroquine. We do have another drug in this category called mefloquine. Mefloquine is specifically useful in treating or combating Plasmodium falciparum. It is tremendously effective against Plasmodium falciparum. Sulfonamides are used to combat parasites, and here we have got two important examples. One is called pyrazinamide in combination with sulfanilamide. These two are used together to combat the effect of parasites. It is used to treat prophylactically malarial infections, as well as it is used for various other parasitic treatments. The second one is called diaminopyrimidine and trimethoprim. These two are also used in combination to combat the parasites. And apart from this, we have got nitroimidazole, which is used to combat amoebiasis, giardiasis and trichomoniasis, wherein we can see that it comprises of two important drugs like metronidazole and tinidazole. These two drugs are used to treat infections caused by amoebias, and the intestinal infections caused by giardia are susceptible to be cured by metronidazole and tinidazole. we have got certain drawbacks that metronidazole sometimes causes certain brown furry tongue, and because of the breakdown of hemoglobin, we can see that some deposition occurs in the tongue. It can also prevent the growth of candida, and it also causes birth defects, and cancer can be passed from infants in the breast milk. So these are the drawbacks of using metronidazole or tinidazole. Next, we have seen the use of heavy metals. They have been used extensively from very primitive days, like the use of arsenic and antimony for the treatment of various parasitic infections. Most important among them is melarsoprol, which is known as MEL-B, is used in the treatment of African trypanosomiasis. Antimonate is also effective against leishmaniasis. So here we can see that heavy metals can also be used to combat the parasitic infections. They actually primarily affect the intense metabolism of the protozoal cells. So herein we have discussed about the mode of action of various antiprotozoal agents. So in our next part, we will be discussing about the mode of action of various anti-Helminthic drugs. Anti-helminths means we are trying to combat flukes, tapeworms, hookworms, which are a bit bigger organisms to combat. So specifically, we use 3 different target sites to combat helminths. Number 1 is the neuromuscular system is to be targeted. Second is the carbohydrate metabolism which is going on within the helminths is the second target site, and third target site is the microtubules. Niclosamides interferes with the carbohydrate metabolism, thereby increases the release of lactic acid. Moreover, we have seen that this tapeworm produces certain compounds to inhibit the action of the host cells. The drugs that we are using here, what they do, they destroy those compounds which are released by the tapeworm to combat the host immune response. Thereby, we can combat the tapeworm. Mebendazole is a broad spectrum antiparasite that is used to treat several roundworm infections. Mebendazole executes its action by targeting the microtubules which are present in the eggs and larvae of the worms. But the problem lies that they may destroy the fetus. That's why pregnant women should not consume mebendazole. It is a chemical compound which is a neurotoxin. It actually attacks the and paralyzes the body walls of the muscles of roundworms. Thereby, they kill the roundworms and they inactivate them. So this is how we have discussed about 3 different ways of combating the helminths. So in this lecture, we have discussed about various antiviral drugs, their mode of actions. Then we have discussed about various antifungal drugs, their mode of actions. We discussed about antiprotozoal drugs, their mode of actions, and finally we have dealt with anti-helminths and their different target sites.